We know that the biggest effect in reservoirs is because of the unnatural fluctuations in the water level, so that the timing and altitude or magnitude, or what is it now, is different from natural. And the organisms that live in this littoral zone, they are really well adapted to this different kind of altitude or magnitude fluctuations in the water level. But in the reservoirs, these fluctuations are much shorter in time and bigger in magnitude. So that makes it more difficult for the organisms to survive. And this littoral zone is the most productive and diverse, usually in the lakes. It's also important for the fish, small fish, for food. And we know that these unnatural fluctuation can have a big effect on these bigger animals that live there, like insect larvae and bigger crustaceans and mussels. But we know quite not so much about the small stuff that live there as well, and can be really important in the sense that they are really abundant, and for the smallest fish they can be the main food source. So in this project we aim for these smallest animals. So the aim is to find out if the regulated reservoirs have a lower diversity than the natural ones, because of these unnatural fluctuations. But because these small stuff animals, they are really small and difficult to identify using morphology, and it takes a long time, we are using DNA here, and we are trying to grasp all of the eukaryotic ones, so that we use really universal primers. And then, because of the aim of the project is this littoral zone, we take sediment samples and do this DNA metabarcoding. And we have two expectations in this, that the biodiversity is either not different in these lakes that have been regulated, or that the biodiversity is lower in these regulated lakes. If there is no effect, then that's a good result. But if it's lower in these reservoirs, we may plan for a bigger project where we can make some estimates how to make the effect smaller. But this is in theory. So in practical work, there will be some difficulties on the way. So the biggest problem here is that we choose the lakes correctly and reservoirs, so that we can take apart the effect of other variables, environmental variables, from the regulation. And our plan is to use 5 to 10 lakes and choose really close by lakes, so that the environmental factors would be as close as possible. But do we find those lakes? That's the main goal here now in the spring, that we search for such lakes that we can take representative samples. Yeah, that was all I had in my mind.